Hello my dear children, Namaste and welcome to yet another session of our Class 10 ICSE series. As you know, we have started with the chapter Genetics, some basic fundamentals, and we had an amazing live session yesterday. All of you participated so well. We have started with the basics of Mendel's experiments, and today we will be continuing with that. Remember, I signed off telling you that Mendel's experiments do not end here. We will be talking more about his experiments in the next session. Of course, yesterday it's not a live session, although yesterday we could manage to make it a lot fun because of your participation. Anyway, doesn't matter. In case you have any questions, any comments, any concerns, do post it in the comment section or do send me an email on my email ID, which I will be showing you at the end of the session. Okay, so introducing myself once again, I am Ambika, Master Teacher of Biology right here at Vedantu. Okay, so let's get started with today's session. This is what I have to tell you first. Children, remember that the struggle you're in today is developing the strength you need for tomorrow. If any of you ever has a feeling that you're getting stuck, you're not able to move ahead, keep this in mind, because don't look at it as a struggle, okay? Because I'm sure all of you have the desire to succeed. All of you at least wish that you could succeed. My sincere suggestion is convert your wish into your desire, your strong, wholehearted desire, and then start working towards it. We've been giving you lots of sessions to keep you motivated all the time. I will be meeting you every Monday for a very short session exclusively to keep you motivated. All the other teachers are again working very, very hard to stay with you and keep you motivated all the time, because we are nearing your board exams. We just have about 3.5 months left before your pre-boards. Let's aim at that, and with that in mind, let's just happily and cheerfully go ahead and do our best. Okay? Now remember, yesterday before we signed off, I had given you 2 homework questions. Not all of you have given me the answer to this. Most of you have given me only the answer to the second question, which was of course a much simpler one, but an important one. Anyway, there was one person who answered this in detail. I will show you the names after we have discussed this. Okay, so I told you that this is a monohybrid cross, and I told you that you have to find out the F1 genotypic and phenotypic ratios, F1 and F2, right? Genotypic and phenotypic ratios for this particular cross. I told you that the character that we are considering here is seed color. Seeds could either be, like in Mendel's experiments, pea seeds can either be yellow or they can be green. And in general, it's seen that yellow seeds are dominant over green seeds, which is why we have represented the yellow seed as an uppercase or a capital Y, and the green seed with a lowercase or a small y. Let us work out this cross. Now again, children, those of you who think that biology needs only memorizing, and if you are fond of physics or mathematics, I'm sure you will find this to be a lot of fun. Just understand the logic of how this works. The only thing you need to keep in mind is that in this cross, we have 2 parents, one which is a pure breed yellow seeded plant and another one which is a purebred green seeded plant. So this will be this genotype and this will be this parent's genotype. What about the gametes? This would produce just one type of gamete, which is this. You could write one more here because to show that the 2 alleles have separated out and entered 2 different gametes, but it doesn't matter here as long as we are aiming at calculating the ratio. We are only concerned about the type of gamete. So now, as you can see, both are the same type of gamete, which is containing the allele capital Y. In this case, it will be the same. It's going to be small y, small y. You can even write it once for this and this. So the F1 generation or the first filial generation would be a combination of these, which would be capital Y, small y, the genotype of the F1 generation. What is the next step? We let it self-fertilize. This undergoes selfing, which means now in our cross, the parents are going to be heterozygous yellow. heterozygous yellow-seeded plants. Okay, the anther and the carpel are from the same plant or the same type of plant, which is why we have represented both the parents as having the heterozygous genotype. Okay, what do we do next? Remember, I told you that we could work out the combinations in a Punnett square, right? So let's see what kinds of gametes are here. For this parent, it would be a capital Y going into one gamete. Another gamete would have the small y. Here again, for the second parent, it's going to be the same, a capital Y and a small y. How would you work this out? Capital Y would be the combination of this, a capital Y, small y here. Here again, a capital Y and a small y, or small y, capital Y, and a small y in this case. These are the different genotypes that we have got in the F2 generation. The F2 generation. Okay, so who's going to tell me what the phenotypes will be? Because there is only dominant allele here, there are only dominant alleles here, this would obviously be yellow-seeded. What about this? It is heterozygous, but still the dominant allele masks the effect of the recessive allele. Again, because of that, this will be a yellow-seeded plant. Again here, these two are the same, one and the same, capital Y, small y, capital Y, however you write it, it's going to be the same. These would again produce yellow-seeded plants. And a small y combination would be an exception because a dominant allele is absent here, and these would produce green-seeded plants. So quickly tell me, what would the ratio be? In F1, you don't have a ratio because all of them were yellow-seeded. Right? In F1, all of them were heterozygous yellow-seeded plants. But in the F2 generation, what do you see? The genotypic ratio will be 1 capital Y, which is this, 2 capital Y, small y, and 1 small y, small y. This is our F2 genotypic ratio. What about the phenotypic ratio? That's very easy, right? You have 3 yellow here and 1 green here. So that makes it 3 yellow is to 1 green. And completely makes sense because these are the yellow-seeded plants. The yellow-seeded plants could have either the homozygous dominant genotype, which is this, or the heterozygous genotype, which is this. The recessive phenotype is always homozygous with the respective alleles. Right? Keep this in mind, children. If there is at least one dominant allele in the genotype, the phenotype automatically expresses the dominant character. For a recessive allele to be expressed, if a plant is producing green seeds, that means it needs to have 2 copies of the recessive allele, which is exactly what we see here. Okay, so this is how you would calculate a monohybrid phenotypic and genotypic ratio. This is exactly the same thing that Mendel came up to as his conclusion. Okay, now looking at this, look at this. This is exactly what we have done now. The F2 calculation in the Punnett square, you have yellow, yellow, yellow. What was this? Small y was green, right? So they are just happy genes, happy peas jumping around. I just thought this would make it a little interesting for you. So coming to the next question that we had. Alleles are found on sister chromatids or homologous chromosomes. Yes, the answer is homologous chromosomes. Remember, children, homologous chromosomes are those chromosomes in which if it's a pair of homologous chromosomes that you're talking about, in the pair you get one member from your mom and the other member from your dad. Okay, sister chromatids are something that you would find only at the time of cell division. They would be attached to each other by a centromere. Remember, in the chapter genetics, we will mostly be talking only about homologous chromosomes. We will not be dealing with sister chromatids, so do not get confused there. I thought it's important to clarify this because as a student, I remember getting confused with both of these. Okay, so clarify this right away in your head. What are homologous chromosomes? Where they segregate, where they pair up, all of those things which we have discussed in our session on meiosis, right? Now, here are the answers that I got. So, like I said, it was only Ayush who gave me the answer to the monohybrid cross. Very good, Ayush. Thank you very much. I was very happy to see a descriptive answer. The only thing was that you have written in the F1 generation will have the genotypic ratio 1:1. Phenotypic ratio also 1:1, which is not the case. Like we have seen, it produces only yellow-seeded plants in F1. Which means all of them are yellow seeded. 1:1 would mean that you have 50% yellow seeded and 50% green seeded. I think perhaps the correct thing was in your mind because your F2 calculation is completely correct. Okay, what you have written as 3:1 phenotypic ratio and 1:2:1 genotypic ratio, and you have written all the allelic combinations correctly. 3 yellow, 1 green, and all of these that you have written here are completely correct, which means I just assume that when you put the F1 thing in the form of a ratio, you somehow went wrong. That's all right, that happens. Keep in mind that when it's all yellow seeded or all expressing one character, you wouldn't be able to write it in the form of a ratio. Your answer would just be all yellow seeded plants and all with the genotype capital Y, small y, which is heterozygous. And we have Isha, Clash with Incredibles, Ronak, Shiva, Adarsh and Siddhi, who have given us the answer for the second question. Alleles are found in homologous chromosomes. Thank you very much and keep it up, guys. Keep up the good spirits. Let us now start our session for today. But before that, let's quickly recap with 2 or 3 very quick and easy questions to see how much of yesterday's session you have understood. Correct? The first question is, the genotype capital T small t is described as homozygous, heterozygous, homologous, heterologous. Give me the answer right now. 4, 3, 2, 1, go. And your answer is heterozygous. When the 2 alleles of a genotype are different, comprising of 1 dominant and 1 recessive form, you call that condition or you describe that condition as heterozygous. Homologous and heterologous are terms that you come across only when we talk about chromosomes, right? The next question. In a pea plant, purple flower color is dominant over white. If a flower is heterozygous for the character flower color, it would appear purple, white, a mix of both, or you think the data is insufficient. Think about it for a bit. Purple is dominant over white and you're given a heterozygous flower. What would its phenotype be? 4, 3, 2, 1, go. And your answer is purple. Heterozygous would certainly mean that the phenotype would be the one that is coded by the dominant allele. And here it's given very clearly that purple is dominant over white. So obviously that will be what is expressed. So yes, remember our hero for yesterday's session, Gregor Johann Mendel, right? We call him the father of genetics because what I would really like to appreciate and I feel very fascinated about when I think of Mendel is that in his times, even nobody knew much about the cells, knew much about the nucleus. Chromosomes hadn't been discovered. Nothing of that sort had been discovered. And he came up with all his theories of inheritance only and only through his careful and close observations, because he was very curious about science. He was very curious about plants, about the science behind plants, about the science of inheritance. He just wanted to know more and more, which made him, like I said, he had that desire, that very strong desire and will to just somehow, come to the conclusion and find out what is it that the mechanism works like in the case of inheritance. How does it work? It was only that strong desire and passion that drove Mendel to success. Like I said, unfortunately, when he was still alive, people did not give him the recognition he deserved. But today we know that he is truly the hero of genetics, which is why we have given him the title Father of Genetics. Now, this is what we discussed yesterday, right? A typical monohybrid cross, which Mendel did for 7 different characters of the pea plant. The example that we chose for our class yesterday was the character height. And remember, we considered tall plants, homozygous or purebred tall plants, and homozygous dwarf plants. The gametes were these, and this is what you got on F1 selfing. All of them were tall, and these are the gametes of the next generation. And yes, these are the F2 combinations that you get. Just like the homework question that we discussed, you get exactly the same ratio, 3:1 phenotypically, 1:2:1 genotypically, and this is how Mendel also preferred to represent it in the form of a Punnett square, because he was also very much into mathematics, and he in fact, we could say that a combination of his logical thinking and his interest and passion in biology, the combination of those two, math and bio, right, have worked wonders. Which is why, in fact, for me also genetics is a favorite topic, because as a student, for me, biology and mathematics were my favorite subjects. I'm not saying that I was an expert in mathematics, but I was good at math, and bio was also my favorite subject. So that way, considering that, genetics has always been my favorite topic in biology. Okay, so look at the Punnett square and look at the phenotypes that you obtain in the F2 generation. And this is where we summed up the genotypic and phenotypic ratios. So I put it here just so that you would be able to link a little better to where we left out. Like I said, it's a slightly concept heavy chapter. It is very, very important that you are familiar with what we discussed in the previous sessions before you attend a session in genetics. Otherwise, you will not understand anything at all, children. So in case you have missed the previous session, do go back, have a look, understand the basics clearly, and then come and watch this again. Things will make much better sense to you. Okay, so let us continue. Based on his observations on the monohybrid crosses, Mendel came up with 2 theories or 2 laws of inheritance. The first one of which is called the law of dominance. Now, what does the law of dominance state? It states that characters are controlled by discrete units called factors. Now, remember, children, I told you that the terms genes and alleles had not been discovered or come up with around Mendel's times. Mendel used the term factors to refer to genes and alleles, both. Okay, so he came up to the conclusion based on these observations that characters are controlled by discrete units called factors. Secondly, he understood or concluded that factors always occur in pairs. Today we call them a pair of alleles which make up one gene, right? And thirdly, Mendel stated that in a dissimilar pair of factors, by which we are talking about the heterozygous case, okay? In a dissimilar pair of factors, one member of the pair dominates the other. Say for instance, in this case, a dissimilar pair of factors, right? A capital T and a small t. Dissimilar pair of factors. One always dominates over the other, which is why these plants are always tall. This is what Mendel meant by the law of dominance. Remember this, children. This is very, very important to know as a basic point in genetics, and also from an examination point of view. What was Mendel's first law, or what was Mendel's law of dominance? Remember, these are the 3 points that you will have to write in your examination if this is asked. Okay, so connect it with the cross that we have written here, and it will make much better sense to you. Just have a look for a few seconds, get it clear in your head, and we will go to the next law. I'm moving away for a few seconds. Okay, children, let us now look at the next one, which is called the law of segregation, otherwise known as the law of purity of gametes. What do we mean by that? Now, only one of the 2 gene copies present in an organism is distributed to each gamete, the egg or the sperm cell that it makes, which is what we have represented here, right? Only one of the 2 gene copies enter into the gametes. Children, ideally, when you represent gametes in a cross like this, it would be better if you represent it in a circle. That is the correct method of representing gametes. So keep that in mind. The first point that we have written here is that only one of the 2 gene copies, we have 2 gene copies here, capital T, and here 2 copies of small t. But during meiosis, when gametes are formed, remember, homologous chromosomes separate out, and it is only one member of each homologous pair that goes into each gamete. Which is why ultimately only one copy of the gene, only one allele, or one of the 2 factors, as Mendel called it, would enter into each gamete. That is the very first point that we have written. Okay. Secondly, the allocation of the gene copies is random. What do I mean by that? I mean that there is no specific factor that decides whether the capital T would go into one gamete and the small t would go into another gamete or anything like that. Especially in this case, in these 2 cases, because they are both homozygous, there is no confusion. All the gametes would get only capital T in this case. All the gametes would get only small t in this case. But in the case of a heterozygote like this, when the gametes are formed, the 2 alleles separate out, and some of the gametes will receive the capital T allele. Some of them would receive the small t allele. They are just random. It's just randomness that decides what allele goes into which gamete. There is no other hard and fast rule for that. Okay, very clear, children? 1 and 2. The third one. When an egg and a sperm join in fertilization, they form an organism whose genotype consists of the alleles contained in the gametes. Very clear, right? Because this is what you can see here. In this case, when the F1 generation is formed, the genotype of F1 is formed by the combination of the gametes that combined during fertilization. The sperm here contains capital T and the egg contains small t, for example. At the time of fertilization, these 2 fuse, making the genotype of the zygote or the next generation a combination of those 2. Again, in this case, you see that these are the gametes, right? This is one parent and this is the other parent. You get all kinds of combinations. Remember, children, be very careful when you work out these combinations. When you have to get the genotype of the next generation, you have to take the combination of one allele of each parent. It will be completely wrong if you took these 2 combinations. It doesn't make any sense. You would get back this. one genotype has to contain an allele from each parent. Look at it very carefully. You get this genotype when you combine this parent's gamete and this parent's gamete. Another possibility is a combination of this and this. The third possibility is this and this. And the fourth one is this and that. Okay? So that is how you have all of the, once again, in the F2 generation, if you look at it, that's how we have arrived at all of these combinations, a combination of these two, a combination of these two that you can see here, a combination of these two, and finally a combination of these two. That is how we get all of these genotypic combinations, which is why the third point says, when an egg and a sperm join in fertilization, they form a new organism whose genotype consists of the alleles contained in the gametes. Very, very important, children. Now, talking about why this law is also known as law of purity of gametes. Now, first of all, why is it called law of segregation? What do you understand by the word segregation? Segregate means separate out. What we mean is that the alleles separate out and enter into individual gametes, which is why we call this law of segregation. It is called law of purity of gametes because one single gamete, if you look at it, is pure or contains only the allele for one particular trait. It contains either the dominant one or the recessive version, right? So you can say that a gamete is pure for that particular trait. A gamete cannot have both the versions of a gene, which is why we also call this law of purity of gametes. So what are the laws that Mendel came up with? Based on monohybrid cross, he came up with the law of dominance and the law of segregation or the law of purity of gametes. Okay? Clear, children? If you have any questions, any concerns about this, do post it in the comments below or send me an email. Do not forget, because this is a very, very important concept. Just mugging up will not work. I'm telling you, understand this, clear it up in your head so that the rest of the chapter becomes much easier for you. Okay? So let us now go ahead with the next one. Now, this again we have seen in the previous session. This is just the table that summarizes or shows all the characters that Mendel chose in the pea plant, right? So the example that we considered in our monohybrid cross was only for the character height, right? Which is why we considered tallness and dwarfness or shortness as the two variations or traits of the character height. So Mendel did such monohybrid crosses for all of these characters, okay? All of these characters, these are the characters he chose. He did multiple experiments based on all of these. After this, in the next step, Mendel was curious to know how things would work if he had to study two characters at a time, inheritance of two characters at a time. That is how Mendel started working on the dihybrid crosses. Remember, we defined dihybrid cross yesterday, the type of cross in which two different characters are studied at a time. Monohybrid would mean one character at a time, right? So dihybrid cross, in the example that we take today, let us consider the characters seed color and seed shape. Mendel did choose different combinations of all of these. For the ease of understanding, we randomly pick two of these characters, which are seed color and seed shape. The variations of this character that you can see are, in the case of seed color, seeds can either be yellow or green in the case of pea plants. Seed shape, they could either be round seeds or they could be wrinkled seeds. Okay, so let us start by working out and calculating the different genotypes. Okay, I'm not using the color green here because green is one of our traits. So let's use red and blue to be on the safer side. So these are the parents that we have, yellow, round and green wrinkled. Again, he chose plants that were purebred for the respective characters, which means they were homozygous for each of these. That is why the parents' genotypes are capital R, capital R, capital Y, and here it's all lowercase or small letters. So in the dihybrid cross, like you see, the genotypes of both the parents are completely homozygous because both are purebred individuals. So let us start by writing the combinations of the parents. The parents are, this is one parent, right? And the other parent is completely recessive. Now comes the trickier part, children. In a dihybrid cross, when you have to write alleles in a gamete, it works different from a monohybrid cross wherein it was only one letter per gamete, right? In this case, in a dihybrid cross, because we are considering two characters or two genes at a time, these are the two genes we are considering here. Each gamete would contain at least one allele of each gene. What I mean is that in this case, it's a capital R and capital Y. R and Y are alleles of two different genes. When I form the gametes of this during meiosis, when homologous chromosomes separate and gametes are formed, each of those gametes would have at least one copy of all the genes in that chromosome, right? Which is why the gamete here would be capital R, capital Y. Right? And in this case, it would be a capital small r and a small y. And thus, the F1 generation would be a combination of these two being heterozygous for both the genes. This would be capital R small r and capital Y small y, but phenotypically, okay, phenotypically, you see that all the F1 plants would be round and yellow. Phenotypically, it is similar to the dominant parent. Genotypically, it is completely heterozygous. Okay? So, keeping that in mind, now let us figure out the next step, which would be selfing of this, which means now the parents that I have both have the genotypes capital R, capital Y, small y, and capital R, capital Y, small y. We are basically selfing the plants. So I consider one as the male parent and one as the female parent, although the anther and carpel can be from the same flower itself, okay, for the ease of understanding. Let us first look at the gamete combinations. For the first parent, it will be, this is one possibility. Remember, one allele of each gene. So I have to have at least one R and at least one Y in every gamete. Okay, this is one possibility. This is another possibility. Small r and capital Y would be the next combination, and a small r, small y would be the fourth one. Similarly, for this parent, it would be exactly the same. Capital R, small r, capital Y, and a small r, small y. Now let us fill up this Punnett square with all of these combinations. It is a combination of these two. So this, the next one will be a combination of these two. The third one would be capital R, capital Y. The fourth one would be capital R, capital Y, and a small y. The next row would be capital R, capital Y, small y, R, both small y. The next one would be these two, capital R, capital Y, and a small y. A capital R, and a small y, small y. Yes, children, are you doing this along with me? I suggest you sit with paper and pen and work out these combinations so that it makes much better sense than just watch me work it out. Okay? and the last row would be this capital Y, small y. The third one would be small r, a capital Y and a small y. And the last one would be small r, and a small y. Take a few seconds, have a look at this, see if it's making sense to you. I'll just move out for a couple of seconds. Okay, children, so this is all the F2 plants combinations that we have. Now, these are all just the genotypes. What do they look like phenotypically is one major thing that we are concerned about, right? Because monohybrid cross, we were very clear about that. It was easy to find out, 3:1. Here, since we have 16 different boxes that we have populated, how many would look like the parents, how many would look different from the parents? We are not sure about that. We need to figure that out, which comes here. Now, I have put this here with color coding so that it will be easier for you to understand at one glance. But what is the orange one or the brown one? It's got nothing to do with the color that it had. It was round green, as you can see. Because it was a combination that is different from the parents, I have put it in different colors. That's the only thing. And these greens are also different. Okay. So the yellow ones that you see look exactly like the yellow parent. The dark green that you see here looks exactly like the recessive parent. The bright green and the light orange or the brownish orange that you see here have a combination of the two, which is round green and wrinkled yellow. Okay, have a look at this and tell me what the ratio is. You would see that the phenotypic ratio would be, yes children, who's going to tell me the answer first? The phenotypic ratio, how many round yellows do you have? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 round yellow is to how many round green do you have? 1, 2, 3. It's only 3 of them. 3 round green is to how many wrinkled yellow do you have? Again, it's only 3, the bright green boxes. 3. And how many wrinkled green do you have? It's only 1 of them. Because of this, Mendel's F2 dihybrid phenotypic ratio is 9:3:3:1. What was a monohybrid phenotypic ratio? It was 3:1. Those of you who are very good at math, tell me if you can see any connection between these two. If you can, that would be great, and do post it in the comment section below or put it in the chats. I will be looking at it when the session is being premiered. What link do you see between the monohybrid F2 ratio, monohybrid F2 phenotypic ratio, and the dihybrid F2 phenotypic ratio, the 3:1 and the 9:3:3:1? Okay? Don't forget, children, this is one of the homework questions that I have for you today. And based on the dihybrid cross, Mendel also came up with one more law, which is called the law of independent assortment. Okay, so it's nothing complicated. It just says that the alleles of 2 or more or different genes get sorted into gametes independently of one another. In simpler words, what we can say is that the allele that a gamete receives for one gene does not influence the allele received for another gene. What I mean is that in the example that we just discussed, right, we have seen that, now in the example that we have just discussed, we have seen that these are the different gametic combinations, right? You see that just because a gamete is receiving a dominant allele for the round or the shape gene doesn't mean that for the other gene also it will receive the dominant allele, right? There can be different combinations of them. Here in this case, if you look at it, this gamete contains the recessive allele for one gene and the dominant allele for another gene. Here again, it's the same. Dominant allele for one gene, the recessive allele for the other gene. Two unrelated genes, but we are studying the inheritance pattern of them together. Which is why this theory says that the alleles of two or more different genes get sorted into gametes independently of one another. Keep this in mind, children. The allele that a gamete receives for one gene does not affect at all, is completely independent of deciding what the other allele for the other gene will be in the same gamete. This is what the law of independent assortment is all about. So we have basically learned three laws that Mendel came up with. Two based on monohybrid cross, which was a law of dominance, and the law of segregation. And the third law is based on his observations in the dihybrid cross, which is the law of independent assortment. Keep this in mind, a very, very important concept. If you have got all of these three laws very clear in your head, do remember to hit the like button right now, children, so that I have understood, and I'm happy that you have got things cleared in your head. Okay, so coming to a summary of what we have discussed so far about Mendel's experiments, okay, in yesterday's session and in today's session. First of all, Mendel is known as the father of genetics. Secondly, gene is the basic unit of inheritance, and alleles are versions of genes. Thirdly, keep in mind that Mendel worked on the garden pea plant, Pisum sativum. The next point I've put is that in his monohybrid cross experiments, Mendel got the F2 genotypic ratio as 1:2:1 and the phenotypic ratio as 3:1. In his dihybrid cross experiments, Mendel got the F2 phenotypic ratio. That's a typo there. It's the F2 phenotypic ratio, not the genotypic. My bad. It's the phenotypic ratio that you get as 9:3:3:1. What about the F2 genotypic ratio? You have to sit and calculate that. That is another of your homework that I'm giving for today, the question of which will come after this. Combinations of genotypes can be worked out and populated in what you call the Punnett square. And from that, you can also identify and deduce what the phenotype would be. Okay? Then, like we discussed last, based on his observations on the monohybrid cross, Mendel formulated his law of dominance and the law of segregation. Dominance and segregation. And based on the dihybrid cross, Mendel came up with the law of independent assortment. Do not get confused between these two, children. They both are completely different. If you really understand what segregation means and what independent assortment means, remember that segregation is only separation. These two are based only on the monohybrid cross. This is based on the dihybrid cross. Keep that in mind during your exam, in your exam hall, in case you're asked to state the law of independent assortment. Remember first that it is based on the dihybrid cross. And if you feel stuck, work it out somewhere, Mendel's dihybrid cross, and see what you observe from that. Link it with the name of the law, and you will be able to explain the theory well enough. Okay? So that is just a small tip that I wanted to give you. And yes, like I said, your homework question is, what is the genotypic ratio of Mendel's F2 generation of a dihybrid cross? Genotypic ratio. Remember those 16 boxes that we populated in the second Punnett square? The genotypic ratio from that phenotypic, yes, we have already calculated it as 9:3:3:1. And I've also given you another homework, which I've not put here as a question. What is the relation between the F2 phenotypic ratio of a dihybrid cross and the F2 phenotypic ratio of a monohybrid cross? Okay, I want the answer to that. I want the answer to this, and I also want the answer to this question. If you have a tall pea plant, how would you identify its genotype, given that capital T and small t are the dominant and recessive alleles respectively. This is slightly tricky in the sense that we have not explicitly discussed this in our sessions yesterday or today, but just think about it, see what you understand from whatever you can find out from your classroom, from your textbook, whatever it is. If you understand that, do explain it very clearly in the comment section below or send me an email with your explanation. If you just get confused, it's all right. We will be explaining this. I will be explaining this definitely at the start of tomorrow's session. But I would like to see how much of all of this you have understood. That's why I think you have to think independently. See if you can answer this. 3 homework questions totally, children. Don't forget, I want the answers, the best answers for all of those right now in the comment section. And yes, what stage of this are you in right now? I want to comment on that too. Which step have you reached today? Yes, I want answers from each of you. We have to keep going up, and by the time we reach December or Jan, our goal will have to be at least somewhere reaching the top. Okay, we have to go up and say I did it finally when your board exams come. And in that journey of going from here to there, I am with you and all of us are together in this journey. So children, do not forget to download Vedantu's learning app. Thank you very much for joining me today. Like you see, the happy genes are smiling at you. Do download Vedantu's learning app. Do stay connected with us through the Telegram group. As you can see, the link is given right below this video. And most importantly, do not forget to hit the like button. If you have enjoyed this session, do share it with all your friends who you think would benefit from this. And please do remember to subscribe to our channel Vedantu 9th and 10th if you haven't done it yet, because we will still be coming up with much more interesting sessions in the days to come. We have planned a lot of things for you so that your board exams can be much easier. In the meantime, do stay connected with me on my email ID ambika.gopalakrishnan@vedantu.com. If there is anything I can help you with, all of you who have been sending me emails already know that I personally reply to all of you. Even if not immediately, within a couple of days, you can expect a reply from me. So stay connected, children. Do post your comments in the comment section below. If there is any specific topic from this chapter genetics that you want explained again, or any new topic from the chapter genetics that you specifically want a session on, do let me know in the comment section below. I will be having a look at it and we can plan the remaining sessions accordingly. And until we meet again in another amazing session, happy studying, and this is Ambika signing off. Bye bye.